Hello everyone, my name is Jahnavi from Andhra Pradesh, India. I am doing the internship under the undergraduate inbound credit mobility program, which is an integration of University Malaysia Kelantan and Angru. And as I am from India, I arrived to UMK Kelantan on Feb 14 to the UMK city campus, where we had an opening ceremony with the UMK staff. And on Feb 15th, we went to the UMK Jelly, and the training was started on Feb 16th. And this is the picture of opening ceremony at UMK, and this is at UMK Kotabaru, and this is at UMK Jelly campus. These are the personal information about myself, and I'm studying the final year BTech Food Technology in the College of Food Science and Technology, Pulvendla, which is under the Acharya Enjiranga Agricultural University. And my project is about the plant tissue culture, especially on the orchid of on CDM. And the location of training site includes this is the plant tissue culture laboratory, and this is the nursery. And at the working site, this is the subculturing in lab, and this is a picture of the nursery and the media preparation at the nursery. And this is the explants in the growth room, and this is the reporting and presentation in the laboratory. Here comes the plant tissue culture. Plant tissue culture. It is a collection of techniques used to maintain or grow plant cells, tissues, or organs under sterile conditions on a nutrient culture media of known composition. It is widely used to produce the plant in a method known as micropropagation. Methods of plant tissue culture. It includes two methods, which is the type of in vitro growth, which has callus and suspension culture, and the type 2 is the type of explant, like from the single cell culture or shoot and root culture, and from the ovule, ovary, etc. And the environmental condition includes three major important aspects, which is the nutrient medium and aseptic condition and aeration of the tissue. Nutrient medium. The medium is generally depends upon the type of plant tissue or cell used for the culture, and the general nutrient consists of the inorganic salts like both macro and micro elements, and the carbon source usually sucrose, iron source vitamins, amino acids, growth regulators, and an optimum pH of 5.8 is very important. And the aseptic condition. The nutrient medium, as the nutrient medium contains the sugar, which increase the growth of microbes. These microbes compete with the growing tissue and finally kill it. So it is very important to maintain the aseptic condition. So sterilization is very important to stop the growth of the microbes. And the aeration of tissues. The proper aeration of tissue is an important aspect. It is achieved by occasional stirring the medium by stirring or by an automatic shaker. The advantages of plant tissue culture includes the production of exact copies of plants that produce particularly good flowers and fruits, and to quickly produce mature plants, and to modify the chemical structure and disease free and desired product, and to get the easy purification of the compounds, and the production of plants from the seeds that otherwise has low chances of germinating and growing, like orchids, nepenthes. Orchids. I was allotted to the orchid, especially the oncidium, which is abbreviated as ONS in the horticultural trade, which has the about 330 species of orchids in the genus from the subtribe of Oncidinae of the family Orchidaceae. And the common names for these plants include dancing or lady orchid and golden shower orchid. The characteristics of the Oncidium species includes the presence of column wings and the presence of complicated callus, and it has pseudobulbs with one or three leaves, and it has the basal bracts at the base of the pseudobulbs, and the flower comes in the shades of yellow, red, white, and pink. And media preparation for the oncidium. Murashik and Skoog medium is a plant growth medium which is used in the laboratories for cultivation. And here for the oncidium, we prepared three medias for especially for multiply, elongation, and rooting, which is generally same for all except in PGR and activated charcoal. In multiply medium, the PGR used is BAP, which is in cytokinin, and in the rooting medium, IA is used, which is anoxin. And the or activated charcoal is used only for the rooting medium. And this is the pictures of media preparation in the laboratory for oncidium. And here comes the subculturing of oncidium. Subculture, it is either a new cell culture or the microbiological culture made by transferring some or all cells from a previous culture to the fresh culture medium. This action is called as subculturing or passaging the cells. Subculturing is used to prolong the lifespan or to increase the number of cells or microorganisms in the culture. And generally it takes place in the laminar under the laminar airflow, which gives the sterile condition for subculturing. And in the laminar airflow, it has the HEPA filters, which gives a continuous displacement of air to remove the contaminated particulates from the air. And this is the pictures of the subculturing, and this is the collecting explants from the growth medium. Subculturing under the laminar airflow chamber. And here comes the acclimatization. Acclimatization, it is the adaptation of plantlets to a new environment. This process generally involves first washing the plantlet under water and to remove and should remove the medium. We should make sure that no roots get damaged here, and we should observe the plantlet. If it is contaminated, we should add the fungicide, and the plant should be placed in the fungicide for 30 minutes, and after 30 minutes it should be placed in the tissue covered box, which should be lived for 1 or 2 weeks. After then it is transferred to the nursery is a place where plants are propagated and grown to a desired size, and mostly the plants concerned are for gardening, forestry, and for conservation biology. And this includes like retail nurseries which sells the general public and also works for the plant breeding. The activities we have done in the nursery includes ginger sowing, pruning, preparing media for nursery plants, tying up the plants, arranging the pots, and planting, watering, weeding, etc. These are the pictures of the nursery management and housekeeping in the nursery, like ginger sowing, weeding, watering, and media preparation. Learning outcomes, like here we gained the knowledge about plant tissue culture, and we had an hands-on experience on subculturing dot sheets, especially on cdium, and preparing three medias for dot sheets like multiply, elongation, rooting, and the acclimatization of plants, nursery management, and housekeeping. And thank you for this opportunity.